Okay, so today we're going to be studying about anatomy of flowering plants. So what do you exactly mean by anatomy? Anatomy is the study of internal structures. It can be anything. Even if you say you're studying the internal structures of plants or animals or anything, it is still called anatomy. For plants, there is no different thing like a name of studying internal structures of plants is also anatomy, and even for animals also it is anatomy. There is no different name. Okay, so this is something we are all familiar with: cells to tissues, tissues to organs, and also there's something, of course, organs to organ systems is also the other thing. But so cell is the basic unit of life. So it's from cells to tissues, tissues to organs. So it is the basic unit for anything. Okay, even for plants it's the basic unit, and even for animals it's the basic unit of life. In case of organs, you have different organs. Different organs have different internal structure. That is obvious, right? Like the internal structure of maybe the xylem and the internal structure of the phloem is completely different, right? And they are what? The vascular bundles, the conducting systems of the plant. They help in the transport of water and food respectively. So yeah, the different organs have different internal structures. So why do they have different internal structures? These internal structures are designed based on the adaptation to diverse environment. Like in case of a cactus and in case of, let's say, sunflower, they live in two completely diverse environments, different environments. That's why both of their internal structures are going to be same. Going to be different, right? Adaptation to different uh, diverse environment conditions. So let's study about tissues. So we all know cells, like basically many cells make up a tissue. So common origin of all, like tissues are basically a group of cells. So common origin for all the cells, and they also perform a common function. So plant is made of different kinds of tissues, which are the meristematic tissues and the permanent tissues. So these are classified on the basis of cells which are formed. If they have the capability to divide or not. Based on that, the cells are like formed. So okay, so different types of tissues are meristematic tissue and permanent tissue. Okay. So now let's study about the meristematic tissues. In case of a meristematic tissue, the growth is usually restricted to certain groups called meristems. Meristems are what are they? Specialized regions of active cell division. Those are called meristems. Okay, so there are also different types of meristems like intercalary meristem, apical meristem, and all that. We're going to learn about all of that. So meristems, so at the tip of root and shoot, they form a primary tissue, and this we call is the apical meristem. The apical meristem, so if it's formed from the root, it's called a root apical meristem, which is abbreviated as RAM. And if it's formed from the shoot, it is called shoot apical meristem, or SAM. Okay, so shoot apical meristem, so root apical meristem is usually the tip of the roots. But in the case of shoot apical meristem, the distant most region of stem axis is where it is present. Okay, the distant most region of the stem axis is where the shoot apical meristem is present. So in the formation of stem and leaves, certain cells are left behind from the SAM. Okay, some cells are left behind from the SAM. So these cells constitute the axillary buds. So they form the axillary buds. So like axils of leaf, they form the branch or flower. So now you'll be wondering, what are the axils of leaf? So here's a small diagram. So this part, if you can see, this part is called the axil of leaf. Okay, we have the stem here, and node is where like the leaf arises from a region of the stem, which is called the node. What is internode? The space between two nodes is called internode. So nodes and internodes are not the same. Okay, so formation of stems and leaves, certain cells are left behind from the shoot apical meristem, and then they constitute the axillary bud, and then like axils of leaf, they form the branch or the flower. Okay, yeah. So merisms which occur between mature tissues is called the intercalary meristem. Okay, so they occur in grasses and the regenerated parts which are removed by the grazing herbivores. They occur in grasses and regenerate the parts which are removed by the grazing herbivores. Okay, apical and the intercalary meristem together they form the primary meristem. Now, why do they form the primary meristem is pretty important. Okay, it's just a one-liner. So basically they appear, the apical meristem and the intercalary meristems, they appear early in life and they contribute to the formation of the primary plant body. So what is the primary plant body? So there are certain regions of the apical meristem, they produce dermal, ground and vascular tissue. We're going to be studying about all these tissues in detail, don't worry. So because they appear early in life and it gives rise to a formation and contributes to the formation of the primary plant body. Okay, what is primary plant body? Certain regions of apical meristem, they produce ground tissue, vascular and dermal tissue. That's all. Okay, so the secondary and the lateral meristem. Secondary or the lateral meristem, so it occurs in mature regions of root and shoot of many plants. Like basically, if you have seen secondary meristem, so basically they're woody in nature. Like they have thick and they have a woody axis like this. So they're somewhat like this. Okay, so occur in mature regions of root and shoot of many plants and their woody axis. So later they appear later than primary meristem. That is something obvious because the name itself says secondary, right? It doesn't say primary. So secondary in the sense you can just think it as later than primary meristem and their cylindrical meristems. This is something a cylindrical shape, let's say that. So occurs in mature regions of roots and shoots of many plants, woody axis. Later than primary meristem, cylindrical meristems. Example are the vascular cambium, interfascicular cambium and cork cambium. So basically you have three: vascular cambium. You should remember all of these, okay? Interfascicular cambium and cork cambium. So all of these three, okay, are responsible for producing primary tissue. Now if you remember, what was responsible for the production of primary tissue? This is secondary tissue. All these three are responsible for the production of secondary tissue. But in case of primary tissue, we read that the apical meristem, right? Basically the meristems at the tip of the root and the shoot, you call them apical meristems. They form the primary tissue. That is, so you should know which is responsible for the formation of primary tissue and which is responsible for the formation of secondary tissue. So primary tissue is apical meristem, which can be root apical meristem or shoot apical meristem, and they form the primary tissue. Okay, in case of secondary meristem, so vascular cambium, interfascicular vascular cambium and cork cambium are responsible for the production of secondary tissue. Okay, so this is about the meristems. What exactly are the meristems? So we have basically primary meristem, secondary meristem, and what are primary meristems, secondary meristems, what are meristems in general is what we learnt here. Okay, so now we're going to go, so now we just studied about the meristematic tissues now, but that's all about them. Now we're going to study about the permanent or the mature cells. Okay, so after division of the primary and secondary meristems, primary and secondary meristems, when I say it's not the meristems which are going to differentiate, it's only a certain cells of the meristems which are going to divide. So then new cells are formed and they become structurally and functionally, become this one, and they lose their ability to divide after the division of the cells of the primary and secondary meristems. New cells are formed, they become structurally and functionally specialized. Okay, this is important, and they lose their ability to divide. As I told you, the tissues are classified into two types. What were they? Meristematic and permanent tissues, right? So here also I told you here itself on what basis are they classified, because cells which formed, if they have the capability to divide or not, that is important. Okay, Now we're going to learn about permanent tissues. So permanent tissues, they are also further divided. So you have simple tissues and complex tissues. So in case of permanent tissues, so all cells similar in structure and function, you call them simple tissues. Okay, so there are many different types of cells in in the particular tissue. Okay, so what is tissue? We all know it's a group of cells, right? So tissue is just nothing but a group of cells. Because a group of cells, so there are certain cells. If all the cells, let's say, are similar in structure and function, they look like, assume all of these round, no? So I can call them a simple tissue. But if I have a complex tissue, there are different kinds of cells, like maybe something like this and all. This is not a really apt diagram, but yeah, I'm just trying to tell you the difference between a simple and a complex tissue. One second. Okay, yeah, that's all. So yeah, many different types of cells complex, and if they're all similar in structure and function, it is simple. Now simple tissues. So simple tissues means only one type of cell, okay, because all of them are similar in structure and function. That's why only one type of cell. So you have, it is divided into three: parenchyma, collenchyma, sclerenchyma. Okay, what are they? Parenchyma, collenchyma, sclerenchyma. Okay, so we're going to deal with each of them one by one. Usually you are asked to know the differences between all of three, hence I've drawn them in columns, and I've also given pictures of parenchyma, collenchyma and sclerenchyma. Okay, let's start with parenchyma. So it is the major component, and cells are isodiametric. Iso means similar. So basically they have the same diameter is what you mean by isodiametrical metric. Okay, so it's the major component. Cells are isodiametric, spherical, oval, round, polygonal or elongated shape. Okay, so spherical, oval, round, polygonal or elongated shape. Then the thin walls, they have the presence of cellulose. These are the important points. Yeah, you should know it's a major component, and you should know what are the shape of these cells, and also what kind of shape they exactly. Isodiametrical. Okay, so there's spherical, oval, round, polygonal or elongated. Okay, so the thin walls, you should remember how are the walls. The walls are thin and they are with cellulose. Okay, and they're closely packed, and they have small intercellular spaces, or sometimes even no intercellular spaces. Okay, sometimes, but usually they have small intercellular spaces, and their main functions are photosynthesis, storage and secretion. Okay, again we're going to go over it. It is the major component. Cells are isodiametric, which means that they have the same diameter or similar diameter. Okay, it can be spherical, over round, polygon or elongated. Thin walls which are filled with cellulose. You have closely packed structures, small or no intercellular spaces. Photosynthesis, storage and secretions are the functions of parenchyma. This is how parenchyma looks like. Okay, in parenchyma. So this is the transverse section, the TS of parenchyma, and this is the longitudinal section, which is the LS of parenchyma. Okay, transverse means it is cut in this fashion. I mean something like this. Transverse means it's cut like this, and longitudinal means it's cut like this. Okay. Yeah, that's the difference between transverse section and longitudinal section. So you can observe it. So very few intercellular spaces, right? No or very few intercellular spaces. Usually they have small amount of intercellular spaces. So these are the intercellular spaces. Okay, so you have the presence of cell wall, nucleus, cytoplasm, intercellular spaces, vacuoles are also present. Yeah, that's all. It's basically the same thing. Just cut into different sections so you can see how they are. Okay, yeah. Now let's move on to collenchyma. In case of collenchyma, so it is present below the epidermis in dicotyledonous plants. So just a homogeneous layer, or it's usually in a layer, or it is present in patches. Okay, so there are thick walls, thick cells at corner. Okay, so the thick cells at corner, cellulose, hemicellulose or pectin. Like it's made of thick cells, and it can be made of any of these materials: cellulose, hemicellulose or pectin. Okay, so yeah, below the epidermis and dicotyledons, it's homogeneous layer, or it can be found in patches, and thick cells at the corner, which is cellulose, hemicellulose or pectin. oval, spherical or polygonal in shape, and the thing is here they contain chloroplasts. But in parenchyma, chloroplasts were not there, right? So they contain chloroplasts. Okay, so intercellular spaces are absent over here. Okay, so but in that there were small intercellular spaces were there. And what is its function? Mechanical support to growing parts of plants such as young stem and petiole of a leaf. Okay, here what was the functions? Photosynthesis, storage and secretion. Okay, so these are the differences between parenchyma and collenchyma. We can go over it again. So major component and all that. So here it's below epidermis in dicotyledons. Here nothing like that is mentioned. And here the cells are not isodiametric, only here it is isodiametric. You have the shape of the cell, oval, spherical, polygonal. Here also spherical, oval, round, polygonal, elongated and all that. Here you have thick walls, here you have thin walls. So at the corner it's only made up of cellulose here, but it can be made up of cellulose, hemicellulose and pectin. Here there are only small intercellular spaces, but here intercellular spaces are absent. So the different functions, this only gives rise to photosynthesis, storage, secretion, but here mechanical support to the growing parts of the plant such as the young stem and petiole of the leaf. So that is about collenchyma. So now let's just see a picture of collenchyma. This is the collenchyma. As you see, there are no intercellular spaces. Of course, even this has a vacuole. You have a cell wall, a protoplasm. Usually if the protoplasm is present, then it is not dead. Okay, then you have thickened corners. As I told you, the walls were thick. So what were the present? Cellulose, hemicellulose and pectin. Here also vacuole is present. That's all in case of parenchyma and collenchyma. Now let's move on to sclerenchyma. In case of sclerenchyma, you have long, narrow, thick, lignified cell wall. Okay, long, narrow, thick. So here also the cell wall is thick and here is lignified. So basically, it has a few or numerous pits. Pits means, like, you know, right? If you go on the street, there like many sort of depressions and all that, something like that. That's what you can think. Pit, and there is another thing which you'll come across this chapter, which is called pith. These two are totally different. Don't get confused between these two. So okay, long narrow cells, thick lignified cell walls. So they are dead without protoplasm, as I just mentioned a few seconds back. So protoplasm, if they don't have protoplasm, they are dead. Okay, so they basically sclerenchyma is divided into two parts. They can be fibers or they can be called sclerites. So let's move on to fibers. So thick walled, elongated, pointed in something like this. Okay, pointed cells, groups of various plants. Okay, thick walled, elongated, pointed cells, groups of various plants. Here sclerites means spherical, oval, cylindrical, thickened, dead cells. Okay, they're very narrow cavities, and an example can be fruit wall. So it is groups of various plants. You have the fibers of sclerites. Okay, so in this case of sclerites, they are spherical, oval, cylindrical, thickened structures, thickened dead cells, and very narrow cavities, fruit wall. Okay, here thick walled, elongated, pointed cells, groups of various plants. Okay, and the support here is mechanical support to the organs, same as this one, but here it supported the young stem and petiole of leaf. Here it just says mechanical support to the organs. So this is an example of, this is fiber and this is the sclerenchyma. This is the diagram. Okay, it is lignin, as I told you, thick lignified cell wall. Lumen it has, and yeah, pit. It has pit. It has numerous pit. Okay, pit is different from pith. When I say pith, it is P-I-T-H. That's a different thing. Okay,